Hello and welcome all to the ZOOMinar on molecular basis of proteinopathies. Today's speakers are Dr. Eitan Lerner from the Hebrew University, Jerusalem, and Dr. Gregory Hudalla from the University of Florida. I'm also delighted to welcome the moderators for today, Dr. Elijah Haas from Bar-Ilan University, and Dr. Anand Pravastu from Georgia Tech. I also would like to welcome Carol Hall. She is a collaborator of Greg. So we have a great panel, so let's get started and over to Dr. Haas.
Okay, good evening from Jerusalem. I'm happy to join this important webinar. I want to be short because I think that I don't have to tell you how good is the speaker because you will hear the talk and that is more convincing than any introduction. But I want to tell you two small stories about Eitan, which will emphasize why I think that Eitan is an excellent scientist, because I believe the most important characteristic of a scientist is a personality. And Eitan has a personality that one has to restrain, not to push. When Eitan applied for the graduate school, the dean didn't want to admit him. And I said, on my dead body, because he is a scientist. And it proved to be the right decision. Then when he applied for postdoc at Shimon Weiss lab, I wrote to Shimon. I told him, I can send you a detailed formal letter telling everything that Eitan did and why I think he is good, or a very short letter. What do you want? He said, send me a short letter. So I sent him a very short letter. I sent him, take him, you will not regret, and enjoy. And he took him, and after 6 months, he sent me a letter above expectations. So this is a characteristic of the quality of the work that Eitan did, and the rest you will hear now. So Eitan, please start.
Thank you, Elisha, for the warm words, and thanks, Rams, for inviting me. So good morning, good afternoon, and good evening to you all. So I'm going to tell you a short story today about what I achieved in the last 3 years in my new lab in the Hebrew University. And so, all right, so since I'm not sure whether I'm going to reach the end of the presentation, I'd like to start actually with the acknowledgements for the people in my lab who did the amazing work you're going to hear about now, especially with 2 graduate students, Sophia Zara and Joanna Zamel from my lab, in collaboration with Penn State University with the laboratory of Nikolai Dokolyan, and in collaboration with my friends and colleagues here in the Hebrew University. So my work is mostly motivated from the relation of intrinsically disordered proteins to proteinopathies. And one famous proteinopathy, so to speak, is Parkinson's disease, that is diagnosed symptomatically and appear later in life. But it involves neuronal cell death, Lewy bodies, and those Lewy bodies include amyloid-like fibrils, mostly of alpha-Synuclein, that shed sometimes toxic oligomers, and we all have the gene, and some of us get sick, unfortunately. So in my view, the 40,000 feet big questions that I would like to answer at some point are, would we be able to detect PD early onset biomarkers, and would we be able to treat PD many years before symptoms? However, let's look at the details of one famous intrinsically disordered protein or natively unfolded protein called alpha-Synuclein that is at the heart of amyloidosis that is related to Parkinson's disease. So this protein, like many ITPs, tends to bind many targets non-specifically, some of which it binds and folds on top. And by that, it may gain specific structures and then specific functions. So to the best of our knowledge, major structure-function relationships are related to its formation of helical forms on top of surfaces of membranes, or its self-associated species that form beta strands, mostly cross beta strands, some of which eventually lead to formation of amyloid fibrils. And when I look at this question, a question mark here, I'm mostly asking myself, is this natively unfolded monomer having all of the conformations that it will later be stabilized in when it will bind its targets? Or is it randomly unfolded like a polymer and the target will induce its folding to specific structures? And that's one question, obviously, which leads to the question of a bifurcation between two major states. But there could be other questions. For instance, can it undergo conformational changes from its helical forms on top of membranes directly to self-associated beta strands structures and the other way around? So before jumping into my results, just a reminder of the many phases of alpha-Synuclein. As an intrinsically disordered protein, the best way to depict it is to show the results of molecular dynamics simulations. This one is coming from the FAIG lab. Basically, it explores many different conformations in space. Once in a while, it has these residual secondary structures, at least according to the simulation. Its NMR structure on top of small vesicles, on top of negatively charged surfaces, gains a helical form. On high curvature, it gains a helical hairpin, and it can extend to one extended helix on top of low curvature membranes. And then one out of the many cryo-EM-based structures of the fibril shows you the different structures it can get on top of fibrils that is highly stabilized. You can see that it has multiple stabilizing hydrogen bonds between beta strands within protofibrils and between protofibrils, which means that it's going to be very hard to take it apart, but it's possible. And then it's important also to remember that all of this is another protein that is synthesized in the cell. And like many other proteins, its fate if it doesn't fold autonomously, it can fold on top of membranes, as we said. But then chaperones can latch on top of it. And if chaperone cannot ubiquitinate it or position it towards the proteasome, they will create those chaperone sequestered bodies, which will end up with autophagy, or they can create liquid condensates, and those can mature into solid condensates that include fibrils. So all of that is related to the energy landscape of a protein that might include all of the structures that it might attain at different states, at different stabilities with different rates. And hence, we can study it in vitro and learn about the structural picture of that protein and its conformations. And maybe we can learn something about its functionality later on, while the different conditions in the cell will just shape the energy landscape, basically the stabilities and the rate constants of these conformations. So the questions that we asked lately are the following. We have a free-form monomer that eventually binds to membranes or to itself. So would its conformation
Sorry, Eitan, you are muted. So it's a mistake, yeah.
All right. So would its conformations in free form be similar to its structure in bound states? And even if the answer is yes, will it have stable structural motifs that will survive long enough to promote function? We asked the question about the most elementary oligomer, the dimer, Does it exist? And does it have specific structure or structures? And I hope we will be able to reach the last questions, mostly relevant to the membrane-bound alpha-synuclein state, and maybe even the effect of specific transition metal ions on it. So, the free-form monomer. So what we did, we simply said the following. We want to get atomic resolution on the conformations of a protein. So in principle, we can do what many in silico studies are doing. We can go and do absolutely theoretical simulations, and it will be fine as long as the force field that we chose is fine. On the other hand, what we can do is we can choose spatial results from sometimes low resolution measurements that cover the whole ensemble and somehow transform them into energetic terms that we can use as restraints, as additional additives to the force field. So we have force field with terms that are theoretical, and now we also have terms that are experimental and that are not local, so that they are really constraining our simulation. Now, one, I don't know if it's a limitation because it's a small protein, but it's better to have a faster simulation than a slower one. So instead of doing a fully blown molecular dynamics simulation, one can have coarse graining of your simulation, either at the atomic resolution, which is what is called coarse grained molecular dynamics simulation, or at the energy functions, which is what is called discrete molecular dynamics simulations. And so in discrete molecular dynamics simulations, you take your energetic terms, you discretize them into these step functions of the energy. And then you have an ad hoc loop here that you try different discretizations until your simulation results, which are shown here in cyan, fit the experimental results, which are shown here in red. These experimental results are taken from the work of Alicia Haas and Asaf Gruppi from their seminal work on the distance distributions of the monomer of alpha-synuclein with extremely small fluorescent dyes, and therefore you don't need to, or you need to make some small efforts in order to model the dyes, while in single molecule your dyes are extremely large and you have to model them into your force field as well. And so not looking at one restraint, but 8 here. These are just 8 restraints on one hand, but on the other hand, these cover non-local distances within the protein and within many parts of the protein. And so this really restrains the simulation. Then we have a simulation that involves also a simulation of the ensemble using replica exchange, and the results, after they are equilibrated, are then clustered. And we have an algorithm that finds the minimal amount of structural subpopulations or clusters of structures, where the criterion is that each cluster should have a small RMSD, and between clusters we should have a large RMSD. And so we find the 8 of those subpopulations of distinct clusters. And here we plot 2-dimensional projections onto each time a pair of parameters, but there are many more parameters to show because these are just the chosen parameters in this case. So for instance, the radius of gyration versus the potential energy or versus the RMSD. But we said that this is not enough. We want to take those structures and we want to verify them against additional experimental results. And so what we did is the following. First of all, we did a CD measurement. So I guess most of you already have seen how a CD spectrum of alpha-synuclein looks like. We analyzed the percentage of each secondary structure in the result, and then we compared it to the superposition of all alpha helices, all beta strands, all turns, and all other structures in the simulated structures, and there is a good agreement between the theoretical and the experimental results. However, this is not too strong, so what we chose to do is the following. We did cross-linking mass spectrometry, in which we use either hetero or homo bifunctional linkers that have two chemical groups that are specific. So either they bind two lysines, as long as they are in the distance of the length of the linker, or they bind lysine on one side and aspartic and glutamic on the other side. And then we cleave that protein in, digest it basically with trypsin or with glu-C into peptides. And then we run the experiment on the mass spec. And aside from the small masses of short linear peptides, we also get masses of pairs of connected peptides. And knowing the chemistries, we can track back which pairs of lysines or which pairs of lysine and aspartates or glutamates were connected within the distance that is covered by the crosslinker. However, this is a very good experiment when you have an extremely rigid complex. Alpha-Synuclein is a very flexible protein, and most probably cross-linking mass spectrometry covers just a subset of the distances covered by alpha-Synuclein. So instead of doing a binary decision on whether a distance existed or not, 100% or 0%, we calculated the probability that the simulated distance distribution of a cross-linked pair of amino acids was within the distance covered by the cross-linker. And those arches that you see here on each cluster is one subpopulation out of the simulation, are the experimental results that we got. And they are shown in a grayscale, where the more black the arc is, the more agreeing it is with the cluster. And so what we basically got more than 100 crosslinks. And what we wanted is to see that there is a full coverage of all structures with those results. And indeed, that's what we get. So this gives us a bit more trust on our simulation results. So great, we have 8 structural subpopulation. And next, what you have is, if you have structures that are distinct and you have a very rich atomic resolution or near atomic resolution structures in the PDB, why not perform a comparison? So here's an example that we found it very striking. Structures of cluster 5 here are very similar, show a very nice spatial homology with the structure that the alpha-Synuclein will get when it will bind the surface of SDS micelles, as was covered by NMR. It's just that it's not exactly the same structure. So you already have a confirmation that we hypothesize will be stabilized on top of the surface of the membrane. Notice that some structures in cluster 1 look very similar to the subunit of the fibril, and so on and so forth. So we have some of the clusters exhibiting structural homology to alpha-Synuclein structures that will be stabilized after it will bind to its target. However, all of that is very nice, but it's not so serious if we cannot prove that at least some of them are stable, right? If those relax in hundreds of nanoseconds, what are the chances to latch on top of their target and use those conformational states in order to drive their stabilization on top of the target? And so you can look into the literature, and there are 2 and actually more, I think, studies looking at single molecule FRET inside alpha-Synuclein that measured the dynamics. And all measurements looking at single molecule FRET basically show that within milliseconds, whatever you label in alpha-Synuclein is averaging out into one shock noise limited subpopulation, that when you check its fluorescence cross correlation, it exhibits relaxations in the hundreds of nanoseconds. That is true in Ferreol's work, and it's true also in the work from the Schuler lab using nanosecond FCS. You can clearly see relaxations of FRET in hundreds of nanoseconds. However, we have to remember that FRET, especially single molecule FRET, is sensitive to distance and distance changes within 3 to 10 nanometer, while this protein is much smaller and it might exhibit structural fluctuations that are smaller than the dynamic range covered in single molecule FRET. And I'm telling you that as somebody who employed single molecule FRET for a lot of time. And so you can use a shorter ruler than single molecule FRET ruler that still gives you information at the single molecule level, and check whether you have slow dynamics or not. So this ruler is basically called protein-induced fluorescence enhancement. Instead of using 2 dyes, we use just one that acts as an excited state molecular rotor. It has a very bright trans state and a very dark cis state. And upon excitation, since its dipole strength is very high for the trans, very small for the cis, upon excitation, you photoselect just the trans state. And then you wait the excited state and between 2 competing processes, either to emit a photon from the trans state or to undergo isomerization into the out of plane intermediate 90 degrees or into the cis state, and then you will not see a photon. Right? So this means that fluorescence lifetime is expected to be low as long as its photoisomerization doesn't have any steric hindrance that influences its rotation. Okay. So, however, if, for instance, you use one labeled molecule with this dye and another protein, and they are close by to the dye, which introduces steric hindrance, then the photoisomerization will be slower, which will result in more fluorescence from the trans state. So the fluorescence lifetime of this dye is sensitive to close surfaces to the dye. So this is another implication of how you do it at the single molecule level. So you have a focus confocal spot, and then single molecules diffuse in and out of the confocal spot. And while they diffuse, whenever they are excited, they start their life in trans, and they might emit a photon from trans, or they might not because of transitions to cis. So this results in a burst of photons from many excitations and emissions that lasts for a few milliseconds, and then separation times between it and other bursts of few seconds, right? And within each burst, we get a few amount of photons. Each photon has its own lifetime after it was excited, its own nanotime. And then we can take the average of those nanotimes, which is proportional to the fluorescence lifetime, at least the mean fluorescence lifetime. So we use single molecule PIFI, and the readout is via mean fluorescence lifetimes. Before we go back to alpha-Synuclein, just as a control, when you take double-stranded DNA and attach this dye, sulfoside 3, to one of its registers, and since the duplex of DNA is quite rigid, it should exhibit a very rigid region, a microenvironment for the dye, and you would expect one type of environment and therefore one type of steric hindrance. And indeed, you see it. And in single-stranded DNA, you also have very flexible polymer that is reconfigurating faster than milliseconds. And then you have an averaged out single populations. All right, but this is far from the case in alpha-synuclein. As you can see, in alpha-synuclein, for instance, when sulfoside 3 is labeling position 26 on the NTD segment, you have 2 or maybe 3 subpopulations, where the short subpopulation is of alpha-synuclein molecules, where region 26 is freely rotating, probably solvent exposed, while the higher the lifetime of the subpopulations, the more sterically hindered those structures are. Basically, this shows you that although the molecules diffuse in milliseconds through the confocal spot, you have slower dynamics between the subpopulations, slower than milliseconds. If they were faster, we would have got a single population that is averaged out. And this is not special just for position 26. It happens on many other positions, for instance, 39, 56. We have more and more of that. So just for you to know, again, a single molecule crosses the beam in a few milliseconds. It may be that each subpopulation that you've seen right now might actually be an averaging out result of faster dynamics that occur in times of between photons. So my postdoc, Paul, has developed this technique lately to quantify within burst dynamics in single molecule FRET. And we're now employing it on single molecule PIFI data. And you can see that the results here are, this time are not bursts, this time are dwells within bursts. Show you that there are 3 states in each of those labeling positions. And with this technique, we can also retrieve the rate constants of the transitions while those molecules cross the confocal spot, right? So for the free-form monomer, we have structural subpopulations and structure models. We have homology to bound alpha-synuclein species. Conformations contain stable local structures. And we hypothesize from all of this that free-form alpha-synuclein monomer conformations may code its later functionalities. Some are stable enough to act as promoters of such functionalities. Certainly not in hundreds of nanoseconds. I would say that the relaxation is occurring in hundreds of nanoseconds for the global structure of the protein, while it has local structures that are undergoing transitions in hundreds of milliseconds. The next step for us was to study the dimer. So we started this game about 2.5 years ago when we did a simple chromatography coupled to multi-angle light scattering. That what's so nice about multi-angle light scattering is that a simple readout allows you to calculate the molecular mass of your species without relying on the hydrodynamics. So we get a direct readout from Rayleigh's theory about the molecular mass. And you can see that on mono-Q coupled to moles, we got a single peak that has this weird molecular mass somewhere between a monomer and a dimer. Okay, so I told Sophie, let's take one of our singly labeled alpha-synuclein, one with a donor, one with acceptor. Let's mix them and go to the fluorimeter and see whether we can capture a FRET signature at concentrations that are 2 orders of magnitude lower than the concentrations that we used in the fluorimeter, in the chromatography, sorry. And so here you see the acceptor excitation spectrum. Focusing on the acceptor fluorescence, you see acceptor direct excitation and acceptor FRET-dependent excitation. And just as a positive control, I told Sophie, let's heat the sample for a day at 37 degrees to see if this minimal FRET signature increases because we create aggregates. And indeed, it increases. Okay, so 100 nanomolar is nice, but we wanted to go to lower concentrations. But regular fluorimeters it's very tough to measure the threat in normal fluorimeters below 100 nanomolar. And so I told Sophie, let's do the same experiment, but this time look at the delayed fluorescence decay of the acceptor after donor excitation. What you see here are normalized acceptor fluorescence decays. And the concentration of donor and acceptor mixtures are decreasing. And you can appreciate that the lifetime is longer at higher concentration and decreases to the minimal lifetime of the acceptor from direct acceptor excitation, even at 3 nanomolar. Just to check that we are on the same page, we did the same thing also in FCS-FRET. In FCS-FRET, you can find the proportional signal from the ratio of the amplitude of the cross-correlation of the donor and the acceptor to the amplitude of the donor autocorrelation. And you can see that as you decrease the concentration, the ratio decreases as well. So this was promising to us. We have a small scale oligomerization in equilibrium within hundreds of nanomolars, and it's mostly monomers and dimers. So I said, let's perform those SM-Phi measurements, but this time, let's add titrate wild-type alpha-Synuclein, which is totally transparent in the microscope. All right, so the single molecules that we will measure are either monomers with dyes or dimers where one subunit is labeled. And I hope you can appreciate that in some of the labeling positions, you almost don't see any change in the heterogeneity of subpopulations, while in others, for instance, in position 140 or position 56, you see a change in the stabilization of subpopulations. And that stabilization is also correlated with the formation of something that is diffusing slightly slower, hence the dimer. So how do we gain enough information to restrain DMD simulations in this case? So what we do is we do heteroisotopic cross-linking mass spectrometry. I guess I should start wrapping up. So what we do is we take a normal recombinant alpha-Synuclein and a 15N labeled heavy protein. And we check that 100% or close to 100% of its nitrogens are indeed isotopically labeled. We mix them and then we cross-link them. Now, among the different cross-links that we have, once in a while we will have a dimer where one subunit is light, one subunit is heavy. And therefore, after digestion, we will have a pair of peptides where one peptide is light, one peptide is heavy, and therefore its mass is going to be unique. We will be able to tell that this cross-link came from an intermolecular link and not from an intramolecular link. Doing that, we were able to get a huge amount of cross-links within the subunit and between the subunits, either using lysine to lysine connections that are within 3 nanometer or less C-alpha distance, or lysine to aspartic or glutamic that are within 1 or 1.6 to be exact nanometer C-alpha to C-alpha distance. So then we devised the discrete molecular dynamics restraints from them. And you can see that for BS3, we use the end-to-end lysine to lysine distance, while for DMTMM, we use the C-alpha to C-alpha distance. And again, alpha-Synuclein is a very dynamic protein. If we will tell the simulation, focus just on the distances covered by the crosslinks, we will bias the simulation to just those distances. We didn't want to do it. We wanted to just energetically favor those structures. All right. We don't want to exclude the longer distances that might exist. And also, we trust more the intermolecular crosslinks than the intramolecular crosslinks, and hence intermolecular crosslinks have a higher, a lower energy than intramolecular crosslinks, because the intramolecular crosslinks are either coming from the dimer subunit or from the monomer in equilibrium, and we don't know that. So doing these simulations, we found those beautiful clusters, and it's very interesting because we get a beta sheet structure in the NTD, and we have a very compact dimer structure that is very stable. And as I told you, we didn't restrain the simulation just for the crosslinks. We just energetically favored it. And hence, some clusters agree very well with the intermolecular crosslinks, while others much less. So let's look at the most abundant structure that we got here, C1. Yes. So here is just to show you that it has this exposed or partially exposed antiparallel beta sheet in the NTD of one subunit. And it also has a bundle of beta strands in a beta sheet in the neck and in the CTD that are also partially exposed. This is very interesting. But even more interesting is, if you look in the literature, especially in the literature that tries to cover the biochemical links between Parkinson's disease and alpha-Synuclein, what you find is that there are many covalent links that stabilize oligomers. One of them is the dityrosine links. And the dityrosine links occurs upon radicalization of at least one of the OHs in a tyrosine. And then if they are in close proximity, they will bind. So this happens actually in oxidative stress or in nitrative stress. So the only thing I wanted to check is pairs of tyrosines in the dimer structures. There is also ample literature on these dityrosinates linked to seeds of aggregation. Very interesting is that when we check tyrosine 125 and tyrosine 125, we don't find any of the clusters reaching any structure with a distance that is in close proximity between OH and OH. We check all of the other permutations, and the only one that gives us something very interesting is when we look at OH and OH of two tyrosine 39s of the two subunits, and only in cluster 1. That shows a very close proximity between those OH groups. So we hypothesize that this cluster is not only compact, stable, and abundant within the other dimer structures, it's also have close proximity positioned tyrosines that upon the oxidative stress might latch on top of each other and will stay in the dimer structure. So all in all, we got structural subpopulations with structure models. We have one compact stable dimer with partially exposed beta sheet structures in the NTT and in the neck segment. We hypothesize that these compact stable structures are might not be related to the exposed neck-induced aggregation on pathway, and that it might be susceptible to dityrosine linkage upon OH radicalization. And so this is the summary of our work. And later on, we are going to perform these measurements also on post-translationally modified alpha-Synuclein. I didn't show you the data on membranes, but we did it again on SDS mice cells. We're moving on to do it at more biomimicking membranes and even biological membranes. And we just started performing in-cell studies, and this has been very exciting for me. And with that, I would like to thank you all for listening to my talk, and sorry for the 9 minutes over.
Thank you very much. Personally, I want to say that it is a great pleasure for me to hear a former graduate student making such nice progress and continuing works that we started many years ago. So that means that the field is alive. And we are open to questions, so I will read the questions. The first question is, Eitan, is there any evidence for tetramer conformations?
So we don't have any evidence at the concentrations that we work. We try to keep below 1.5 micromolar. And we try as much as possible to concentrate either on freshly prepared alpha-Synuclein or on a protein that we prepared again or we refurbished. But once in a while we get, for instance, in sec mols, an additional peak that comes before the main peak of aggregates. So they include many types of aggregates of many types of sizes, but none of them might be compared to a tetramer, at least from my experience.
Thank you. The second question, why is a dimer only observed on ion exchange? Is it because of the high salt concentration? Other groups have not found any dimer by standard purification.
Let me take you to our additional results. So, right. So we did also other chromatography coupled to mols, and it also shows up in sec mols as not a monomer, nor a dimer, somewhere in between. We also basically played around with the salt concentrations in our buffer when we did the bulk fret experiments, and we always have a signature of fret at concentrations that are 1 or 2 orders of magnitude lower than the concentrations in which we perform chromatography. Why? I'm well aware of the fact that the dimer question on alpha-Synuclein is highly debatable. There are groups that indeed show that there is no dimer. On the other hand, there are other groups. For instance, the group of Nikolai Dokholian, even before we started working together, who show that it exists in AFM and in many other measurements. Basically, a discussion on that will come in a week or 2 on bioRxiv. So I urge you to read it.
Thank you. Another question from Ifat Miller.
Hi, Ifat.
Thank you, Eitan, for the excellent lecture. Do you know what are the primary nucleation interactions of the dimerization? What are the driving forces for the nucleation?
Great question. So we still don't know that, since our simulations are not looking at the dynamics like regular molecular dynamic simulations do, where you have trajectories. In our case, we also do replica exchange, so we don't have continuity between molecular dynamic steps, right? Because we shuffle between different temperatures and different heat capacities, just to be able to also simulate the ensemble. So I can't answer your question right now, but we are planning also to do some non-equilibrium experiments, and hopefully next year they will come up and we can discuss that afterwards. But the solution is you have to perform non-equilibrium experiments, hopefully with an infinite acquisition time, and obviously the ones in the crowd that are hearing that might say, oh, continuous flow experiments. Yeah.
Okay, now another question. What was the starting model for your MD simulations, and do the results depend upon it? Also, have you considered using beta wrap-ins designed by Wolfgang Hoyer to stabilize the misfolding intermediates and block their further aggregation into amyloid. You can see it in the chart.
So, okay. I guess I can't see it in the chat.
Ah, you don't?
No.
It's in the Q&A.
Oh, okay.
Ah.
Directly to the Q&A.
Ah, okay.
Right. Oh,
yes.
Olga. All right. So the starting model of our DMD simulations were fully extended alpha-synucleins that were apart from each other. And we basically allowed the simulation to equilibrate until we find first contacts. And we did it at many different conditions, and each condition we found different initial contacts. So although we got different condition contacts in each simulation initiation, we got the same results, or very similar results. We checked that. That's one thing. And the second, have you considered using beta rapins designed by Wolfgang Hoyer? No, honestly, we didn't try that. And Olga, you're just one floor above me, right? So we can talk about it later, I guess, because I'm not aware of this.
Now, the next question, as you can see it in the questions Q&A.
Mm-hmm.
Oh, okay, no, it's an answer.
Yeah, it's from Nikolai. Thank you, Nikolai.
May I ask you another question? Can you envision a non-equilibrium experiment in which you let the ensemble relax and see what are the first structures that you can detect, or first interactions or biases from the random coil simulation situation?
So I think that, like all other proteins, chemical denaturation brings proteins to something that is close to a polymer, to the random polymer depiction. And maybe what makes the difference between a random polymer description of a protein and an IDP is exactly that. So one attempt that can be done, but this is mostly in bulk experiments, would be stock flow experiments like you usually do in well-folded proteins, where you follow those residual structures that are forming in the first steps of kinetics. However, the dead time limits you to millisecond kinetics in stock flow experiments, and I suspect that the kinetics is going to be faster than that. And so Ben Schuller developed this very cool microfluidic mixer that is able to perform microsecond dead time mixing, and then along the pathway of the microfluidic mixer after the mixing, you have homogeneous mixing, and then you can follow the single molecule fluorescence-based histograms at different steps. And those steps have been proven to be within few microseconds. So I would say a mixing of guanidinium-induced unfolded synuclein that abolishes any dimer formation, I think, that is labeled with a lot of unlabeled protein, and mixing it with buffer. So that would be my hunch, but we're going to try it anyway.
Okay, another question. This protein does not collapse. It remains as a random coil to some extent, somewhat biased random coil. And what's the difference between these kind of proteins and the folding proteins?
So true, it does not collapse. But on the other hand, it also is not, at least from our results, a random polymer. And so instead of having a very deep energetic well for the folding collapse, you have minor energetic wells for multiple conformations. And until recently, and you've seen the literature that I brought, basically cherry-picked from the literature about the relaxation times. Until recently, people thought that the global structure is undergoing those conformational changes between those potential wells of marginally stable structures in hundreds of nanoseconds. That's true when you look at FRET that sees the whole structure changing. But you can focus on structural dynamics of local structures, and that's what we did. And this started this whole expedition because this was very weird to us, that we got extremely slow dynamics when it comes to the local structures. So to answer your questions, I think the best model would be that you have a very floppy global structure with local structures that are undergoing conformational changes, but much slower. Yeah.
Okay. Do we have time for?
Maybe we'll go to the next speaker, and then we'll take the question at the end. Thank you very much.
Thank you, Eitan, very much, and I enjoyed your talk very much.
Thank you so much. Sorry.
Okay.
All right, shall I introduce Greg?
Yeah, Anand, go ahead. Yeah.
Thank you for the wonderful talk, Dr. Lerner. I'm here to introduce you to somebody who is very different. In fact, Dr. Hudalla, my collaborator, does not study proteinopathies as far as I know. In fact, I don't believe that he studies pathologically related protein aggregation. In fact, he comes at it from a very, very different perspective. He is a chemical engineer by training, at least his bachelor's degree is in chemical engineering. He has master's and PhD in biomedical engineering from University of Wisconsin. Then he did a postdoc with Joel Collier, and he studied how to use peptides to actually form nanostructures that could be used for immunology or vaccination. So he'll tell you. In fact, in general, Greg's lab is interested in the design and control of peptide nanostructures for biomaterials. And he very much comes at it from a biomaterials perspective. So to give you a few examples, he has studied controllable degradation of peptides assemblies. He's looked at triggered assemblies using a peptide to denature a protein and subsequently form amyloid-like structures. In fact, he's one of the first people who recognized the concept of co-assembly. In other words, two peptides that don't self-assemble but would recognize each other and form an amyloid-like structure if they interact. He has also looked at the use of peptide assembly for enzyme immobilization. He can make nanoparticles and microparticles containing enzymes with very little loss of enzymatic activities. And today he's going to tell us about glycosylation, the addition of carbohydrates to peptides to affect structure and activity. So I think most of us know that nature uses glycosylation, but as far as I know, nobody has actually tried to design biomaterials with that feature. So with that, I will let Greg tell you the details.
Great. Thanks, Anant, for the really kind introduction. That surveys a lot of what we've done. Give me a second here to switch over so that my screen pops up. All right, so can you all see my title slide? Yeah. Yeah, great. All right, so yeah, so what I'm going to talk about today So there was a bit of chat happening before some of this started with two of my really close collaborators, Anant, who so kindly introduced me, and Carol Hall, who's also in the audience today. We've worked together for many years on peptide co-assembly, understanding some of the basic biophysical aspects of peptide co-assembly, modeling the structures and studying the structures that form. And I'm actually not going to talk about a lot of that work today. What I'm going to focus on today is an area that I've been working in for about the last decade or so. Started as a postdoc and carried into my independent career, which really considers the opportunity and in turn the effects of adding glycans as chemical constituents onto fibrillizing peptides. And so before we get into the nuts and bolts of the science, let me first please the legal team at UF. So I am a founder of a company, and I have now told you all about that. So the other thing that I want to do before we get into this is really acknowledge the drivers of this work. And so there have really been two scientists in my lab who have carried this work forward. The first was Dr. Antonietta Restuccia, who was the first PhD student in the lab. So she really picked up on this work that I had initiated as a postdoc and really carried it forward for the first years that the lab was really operational. This was really our flagship project right out of the gate. And after Antonietta left, this project was picked up by Ran Zhuo, who is a research scientist in the lab. And this project has been funded by both the NIH and the NSF over the last few years, and we are eternally gracious for the support that they've provided and the opportunity to build this system. Right? And so what I want to talk to you about today is the post-translational modification of glycosylation. Right? And so as Anant mentioned, glycosylation is really common. About 50% of all proteins expressed within humans are modified with at least one glycan appendage. So it's an incredibly abundant post-translational modification. And it's increasingly appreciated that glycosylation alters protein form, right? And so here's one example, mucins, proteins we've been hearing a lot about in pop culture and pop media recently due to their relationship to the ongoing COVID-19 pandemic. And a key feature of mucins is that they adopt elongated structures, right? And this elongated structure emerges because really the core of mucins is characterized by these dense serine, threonine, proline repeats that are abundantly O-glycosylated. So all of the red dots on here on this cartoon are indicative of putative sites of O-glycosylation of mucins. And so introducing or the presence of these appendages dangling off of the side chains of the amino acids has a profound effect on the structure that that mucin molecules adopt. Right? And this is very elegantly recently demonstrated in the most rigorous of scientific communities on Twitter. Right, there was a recent tweet that came from the Chimera X group at UCSF, in which they took the mucin 17 sequence and they put it into AlphaFold. And they asked AlphaFold, "Are you any good at predicting the likely structure of the protein?" Right? And what was spit out by AlphaFold was an absolute mess. Right, unlike the elongated structure that's common to mucins, what instead was observed was this sort of knotty and aggregated protein structure that looked nothing like the native counterpart. And the reason for this is that none of the glycans that are present natively on mucin 17 were present in the model, right? So the impact of the glycans on the structure was completely ignored in the AlphaFold prediction, right? And so here's just one of many snapshots of the effects that glycosylation can have on protein form. Right? Glycosylation also has profound effects on protein function, right? And so here, for example, is a cartoon showing the presence of sialic acid terminated glycans. These red or purple diamonds here are representative of sialoglycans. And sialoglycans, shown chemically here in the middle, are abundant on the cell membrane, right? And among other things that sialoglycans do, sialoglycans are recognized by a family of proteins known as Siglecs. Siglecs can interact with sialoglycans in cis, so same Siglec interacting with the same sialoglycan Siglec acting with sialoglycan on the same cell surface, right? Or they can engage in trans, right? So Siglec on one cell surface engaging with sialoglycan on a neighboring cell surface, right? And this precise and specific molecular recognition event ultimately controls immune cell activation. And so in general, sialoglycan-Siglec interaction tamps down immune responses, shuts down immune cell function, right? And this is eloquently demonstrated in a paper from the Bertozzi group a handful of years ago, in which they demonstrated that NK cells, so a type of immune cell, is capable of killing a target cell that is lacking sialoglycan. So here you have your Siglec, you have a protein on your target cell surface that's lacking sialoglycans. An activating receptor on the NK cell surface is able to recognize its cognate activating ligand. This turns the NK cell responses on, and this induces killing of the target cell, right? And what they demonstrated through cell surface glycoengineering is that by reinstalling Siglec binding glycan, so they demonstrated this synthetically, but then also coupled this to demonstrations that this is a pathway by which tumor cells evade immunological recognition. So tumor cells are oftentimes hypersialylated. This overabundance of sialoglycans on the cell surface can lead to downregulating of NK cell function, right? So there's the first sort of connection between synthetic system and an apathy, not quite a proteinopathy yet, but we'll draw some connections there a little bit later, right? And so where our group has long had an interest, and where a lot of this work started, is an interest in glycan binding, a family of glycan binding proteins known as galectins. And so there are 15 total galectins within the family member. Most of them are characterized by having 2 carbohydrate binding domains, shown here as the sort of open Pac-Man mouth. There's one outlier that we're interested in, but I'm not going to talk about today, which is galectin 3 that forms higher order oligomers. Right? And galectins bind to glycans on the cell surface, in particular beta galactosides that are found both on the cell surface and throughout the extracellular matrix. And the multivalency of glycans leads to intracell, intercell, and cell extracellular matrix cross-linking events that affect different cellular processes, right? So things like adhesion of cells to each other or to the matrix, the formation of what are referred to as galectin glycoprotein lattices, so regulating the transport or translation of glycoproteins across the cell surface. And galectins can also cross-link proteins, leading to the onset, either the upregulation or downregulation of signal transduction events, right? We and others are interested in galectins because of their context-dependent bioactivity, right? So galectins bind to glycans that are found on every cell within the body. And so with that, right, the presence of galectins in any tissue can have some effect on cellular processes. And so galectin-glycan interactions have been associated with developmental processes and immunity, which we view as typically positives, right? But have also been associated with biological or pathological negatives, so things like cancer and viral infection. Right? And so there's a longstanding interest in both understanding how galectins bind to glycans and being able to manipulate these events for therapeutic needs. Right? And so with that, right, to sort of bridge the gap, it's important to acknowledge, so I already talked about the fact that galectins are primarily dimeric in nature, having 2 or in some cases more carbohydrate binding sites, right? So it's important to acknowledge the impact and importance of multivalency in lectin-glycan biology. And so when I refer to multivalency, I'm talking about systems that have more than 2 units, which would oftentimes in biological systems either be more than 2 copies of ligand, 2 or more copies of ligand, 2 or more copies of receptor, right? And in general, in the context of multivalency, we see that the whole is greater than the sum of the parts, right? So it's not just additive of unit 1 plus unit 2, but rather there's a synergy that can build on and extend beyond this, right? And so multivalency, or the establishment of multivalent structures, can often provide a checkpoint in signal transduction, so ligand-mediated cross-linking of receptors can drive their activation, multivalency can also strengthen binding through what we refer to as avidity effects, right? So increasing the probability of ligand-receptor interaction. And so it should come as no surprise that multivalency is central to galectin-glycan interactions, right? And so where this work really started for us is that we use peptide-based platforms, particularly peptides that assemble into fibrillary structures, to mimic multivalent galectin-glycan interactions, right? And so this platform is really based on a synthetic peptide that I've been working with since my postdoc, which we refer to as Q11, shown here in green. It is an 11 amino acid peptide that will form beta sheet nanofibers in water. And the modification we made is to append a single monosaccharide, N-acetylglucosamine, or GLICNAC, onto the N terminus of Q11. Right? The concept here is that in water, Q11 will self-assemble to form these elongated nanofibers shown here in cartoon, here in this very dense electron micrograph. I'm sorry, I'm not sure what's going on. I seem to have some automation that's taking control of my slides. The reason why we like this nanofiber system, right, is that others that had come before me had demonstrated that we can either tune glycan density, so we can co-assemble the glycosylated variant with the non-glycosylated Q11 to control how much sugar is there. Right? And what my work really drove forward was showing that not only could we introduce monosaccharides, but we could start to build oligosaccharides as well. So here's the transition from this mass spec shows the conversion of the monosaccharide GLICNAC into the disaccharide LACNAC, or N-acetylactosamine, which is a galectin binding disaccharide. Right? And so what we then did with these material systems was ask the question of whether or not we could disrupt galectin cell surface glycan interactions. And so the model that we use is based on T cells, which express membrane glycoproteins that can be bound by galectin-1 or galectin-3. This galectin binding event induces changes in T cell behavior that include agglutination, which is the aggregation or clustering of the cells, apoptosis, which is programmed cell death, and in some cases, cytokine secretion. Right? And so building toward this, we first started with the LACNAC Q11 nanofibers. We demonstrated that by immobilizing the disaccharide on the nanofiber in this multivalent organization, that you can increase, that this would lead to increases in the binding affinity for galectin-1. So the red trace here is binding of galectin-1 to the nanofibers, and the black trace here is binding of the free sugar, the soluble sugar to galectin-1 in solution. So it's been an order of magnitude increase in the binding affinity in this system. Right? And what this increase in binding affinity leads to is an increase in the effectiveness of the nanofibers for disrupting galectin signaling. Right? So you see here in this graph, nanofibers with increasing density of LACNAC, ranging from no LACNAC all the way up to 25% LACNAC in nanofibers. You see that as the amount of sugar in the nanofiber increases, cell viability also increases. So we're able to inhibit the cell death that's caused by galectin-1. Right? And what was really exciting for us at the time is that this inhibitory effect was more pronounced than a stable variant of lactose that was then being pursued in clinical trials known as TDG, which was ineffective at inhibiting this response at a 2-fold molar excess relative to the sugar concentration on the nanofibers. Right? And so one of the next questions that we asked, which was enabled by the simplicity of how these systems are built, is how do features like glycan density relate to the binding affinity? And so here Antonietta used a model in which she systematically varied the concentration of glycosylated Q11 and non-glycosylated Q11, asking, is there sort of a sweet spot, right? So if we control the valency of the sugar along the nanofibers, can we minimize steric penalties while maximizing avidity effects? Right? And so what she observed in the very first experiment was that by diluting the sugar out, so this is 250 parts GQ11 to 750 parts Q11 relative to 250 parts GQ11 on its own, that the nanofibers with the diluted sugar bound to a model lectin, in this case wheat germ agglutinin, more rapidly than sugars with the dense, the nanofibers with the dense sugar along their backbone. Right? And by digging into this a little bit, using a pair of experiments where on the left, the density was varied and the valency was fixed. So the GQ11 concentration was held constant and the Q11 diluent amount was varied. You see that maximal binding was observed at sort of this mid-range, so where the sugar was diluted by the presence of Q11. Right? In a subsequent experiment where both density and valency were varied by controlling or changing both the GQ11 concentration and the Q11 concentration, you see that this sort of moderate sugar density leads to the greatest extent of binding, and that even if you increase the amount of sugar fourfold, that the binding is not as pronounced when those molecules are immediately adjacent to one another. Right? And so what this leads to is a further increase in the inhibitory efficacy of this material system. So here again, we're looking at a T cell death measured as metabolic activity assay. And you see in black here, nanofibers where the glycan is diluted along the backbone are more effective at inhibiting T cell death than nanofibers in which the glycan is densely packed along the nanofiber surface, right? So this is for us a demonstration, right, that the physical presentation, the way in which these glycans are presented in this multivalent context has a pronounced effect on lectin-glycan interactions. And that if we go back a little bit, right, that may not come as some surprise. If you think about the size of a beta sheet strand and the size of a single sugar relative to the size of the proteins that they're binding, right? As is overemphasized by this cartoon, binding of one protein to one sugar in a very dense glycan environment is likely to cover many glycans, rendering them inaccessible to other protein molecules in the system. Right. And so building on from this, so this is a demonstration of tuning the strength of the interaction, optimizing the strength of the interaction from a physical sense. Right. And so one of the other questions Antonietta asked was, can she improve upon the system through chemical means? So in particular, can she encode selectivity or specificity for target binding by manipulating the chemistry? And so to do this, she synthesized an alternative disaccharide variant known as lac-di-nac, in which the galactose is replaced for gal-nac, so an acetylated counterpart. Right? And the idea is that historical data had suggested that this was a specific glycan for galectin-3, but would not bind to galectin-1. Right? So what she observed is that nanofibers displaying either lac-nac or lac-di-nac were both capable of binding to galectin-3, whereas only nanofibers displaying lac-nac, shown here in the open circles, were able to bind to galectin-1. There's very little to no binding of galectin-1 to the lac-di-nac nanofibers. When she took a solution that had both galectin-1 and galectin-3 present, as shown here in controls, galectin-3 is the upper bar, galectin-1 is the lower bar. You see that galectin-3 is depleted where galectin-1 is maintained, right? So there's a selectivity even in the presence of a very similar protein partner in this system. And so chemical change coupled now with physical change should allow for optimization of the efficacy of this material system, right? And so to wrap this all up, demonstrated that these nanofibers are also capable of, in this case, inhibiting galectin-3 signaling activity. So again, using the T cell death assay, we see that cells survive better in the presence of nanofibers than in the absence of nanofibers. And maybe more strikingly, you see that cells in the presence of galectin-3 and absence of nanofibers undergo agglutination, whereas the cells in the presence of both galectin-3 and the nanofibers remain in the single cell suspended state that you would expect for healthy T cells. And there's an aside here. So I have a couple of stars and some red text down here to note that one of the things that we observed in the course of these studies that we didn't expect was that serum glycoproteins that are present in our cell system that can bind to galectin-3 act as competitive inhibitors, right? And these are really abundant serum glycoproteins like IgGs and alpha-2 macroglobulin. And what we think is one of the longstanding problems in galectin inhibitor design is simply the sheer number of targets that galectins have to bind to in vivo, right? And so in these studies, we demonstrated that by depleting the availability of these serum glycoproteins, you could amplify the effect of the material system. So for those who are interested in the inhibitor design side of things, I encourage you to go dig into this a little bit deeper, right? In the paper that was cited there. where we're moving now, right, and this is a snapshot of some unpublished work, is we're beginning to move beyond disaccharides, right? So we're building enzymatic reactions that will allow us to synthesize more complex oligosaccharides on the surface of these nanofibers and begin to push them in other directions. And I don't really have much beyond sort of some snapshots here of what may be possible in the directions that we're going. But what I want to do with the remainder of my time, right? So up to this point, I've largely talked about function, right? How does glycosylation lead to new function in these systems? What I want to talk about is our work over the last couple of years, now moving in the direction of structure, right? And so where this study starts is that during the course of Antonietta's studies, while looking at glycosylated Q11 nanofibers with a transmission electron microscope, she started to observe some really unusual phenomena that we're going to dig into in just a minute. But one thing to note, the reason why peptide nanofibers like this have long been of interest on the biomaterial side is that at sufficiently high concentrations, the nanofibers will entangle with one another to form physically cross-linked gels, right? And so this is, for those who have experience before, this is like a gelatinous dessert, right? Jello, as it's known in the States, the sort of hydrated, sort of gummy kind of pasty material. So the peptide nanofibers will form a very similar type of architecture. And one thing that we noticed, that Antonietta noticed with the glycosylated peptide nanofibers in particular, was that when viewed between cross polars, there was strong birefringence, suggesting long-range order within this system. And when she looked at the structure of the nanofibers in these crowded environments using both cryo-TEM and cryogenic scanning electron microscopy, she saw a strong degree of directionality amongst the fibers in this system that's relatively uncommon, right? And when she took the glycosylated variant and deglycosylated it or modified the sugar in other ways that I'm not going to dive into, this alignment in sort of hierarchical order behavior was gone, right? So it suggested to us that it was dependent on the presence of the sugar within the system. And so going back to our synthetic capabilities, she created nanofibers that had either LAC-NAC or LAC-DINAC, two very similar disaccharides that only vary in terms of their C2 chemistry. And you see here that the LAC-NAC nanofibers form bundles of many, many nanofibers that are highly ordered and tightly associated, whereas having this acetylated counterpart at C2 completely disrupted this event. And then there's, in the paper that's cited here at the bottom, there's other data demonstrating that this depends on the density of the sugar along the nanofibers. So collectively, for us, this was a striking demonstration that glycosylation doesn't just have an effect on function, but it can have some effect on structure in the context of synthetic peptide materials, much in the same way that glycosylation affects structure in the context of proteins, right? And so this led us to a question that we're actively pursuing now, which is, to what extent does glycosylation act as a structural determinant in peptide fibrillization, right? Or in other words, to what extent do glycans impart new structures in addition to endowing new functional capabilities, right? We're not the only folks to work in this space. So here's where the proteinopathy data will come in, and I'll connect back to Dr. Lerner's talk a little bit on the next slide, but preempting this, right? It's long been understood that glycosylation within the beta sheet fibrillizing domain can disrupt fibril formation, right? So this is a cartoon from a paper published by the Imperiali group in 2003, demonstrating that prion formation, which depends on breakage of a disulfide bond in a reducing environment, is suppressed when the prion precursor is glycosylated. And this is because the presence of the glycan stabilizes this disulfide bond formation. So it prevents the unfolding of the protein that is upstream of the aggregation event, right? More recently, there's been a really compelling body of work from the Pratt lab at USC demonstrating that glycosylation of alpha-Synuclein also acts as a disruptor of its fibrillization, right? And so here they synthesized synthetic variants of alpha-Synuclein that were glycosylated at different points, either single site of glycosylation or multi-site of glycosylation. Right? And what they demonstrated is that glycosylation disrupts the fibrillization process. In some cases, it introduces a longer lag phase, as in the case of S87, right? Whereas in other contexts, it completely knocks out the fibrillization event. Right. And the thing that I want to note about both the Imperiali lab studies and the Pratt lab studies and many other studies that have followed is these are contexts in which glycosylations were present within the beta sheet fibrillizing domain, right? So immediately adjacent to or on amino acids that are present within the beta sheet rich structure, right? Where our systems deviate from this is that glycosylation is present on an appendage that's outside of the beta sheet fibrillizing domain, right? So a couple of years ago now, we started asking what we think is a pretty basic question, right? How far away from a beta sheet fibrillizing domain can a glycan be, where it still has an effect on the fibrillization process, right? So in particular, we went back to our GQ11 molecule, Q11 domain here, a short flexible linker, and this N-terminal glycnac moiety, and compare this to the fibrillization behavior of a non-glycosylated counterpart, right? This work was published earlier this year, but the takeaway is that when viewed microscopically using thioflavin T fluorimetry, you see it, whereas the non-glycosylated molecule forms flocculate in neutral buffer and these sort of reticular structures in water, and the glycosylated molecule formed much smaller structures in buffer and no discernible microscale structure in water. Right? And when we looked at the rate of assembly of these molecules using thioflavin T fluorimetry, we saw that the assembly profile of the glycosylated molecule in neutral buffer, shown here in black, and the non-glycosylated molecule in buffer or water were very similar, right? Whereas the glycosylated molecule in water had this pronounced lag phase, right? There's a very short, not really experimentally discernible lag phase in the NQ11 system, whereas a lag phase in the order of hours in the GQ11 system. And so we've begun digging into this in a little bit more depth, right? And what we've observed is that whereas almost immediately after the hydration step, in the samples of NQ11, you can find non-fibrillar aggregates, you can find some fibrillar species, and a number of protofibril species as well. By the time you get as early as 1 hour after incubation at room temperature, you start to see emergence of more elongated fibrillar structures in the NQ11 sample alongside non-fibrillar aggregates. By the time you get to 24 hours, it's mostly long fibril species that exist and some non-fibrillar aggregates coexisting alongside those extended fibrillar species. Now we counterpoint this with the glycosylated molecule. Now remember, these two peptides are exactly the same, with exception of one N-linked monosaccharide on the side chain of an asparagine that's all the way out at the N-terminus of that sequence. You see that at T equals 0 hours, there were no observable fibril structures in this sample. There are some non-fibrillar aggregates, as well as some protofibril species. By 1 hour, the system is dominated by protofibril species and non-fibrillar aggregates. By the time you get to 24 hours, it's almost exclusively protofibrils that exist within this system. So we're seeing distinct changes in the morphology and the evolution of the nanoscale structure of these species, conferred simply by the presence or absence of a single monosaccharide outside of the beta sheet fibrillizing domain. Right? And so the thing to note with comparison of phosphate buffered saline, which is our buffer of choice relative to water, is that deionized water, is that the saline is at pH 7.4 and an ionic strength of about 150 millimolar, whereas the water is at pH of about 5.5 and is reasonably considered to be deionized, right? And so these are two variables that could be affecting the overall assembly process that have changed when we transition from PBS to water. And so we dug into this through a series of experiments in which either one of those variables was varied. The work on ionic strength is in the paper. Nothing significant there. Nothing changes when the ionic strength changes. So it's not the ions that are driving this. But what was unexpected and remains an area of active consideration for us is a pH-dependent effect. So I'll start with a graph on the right. These are kinetic growth profiles for the NQ11s, the non-glycosylated peptide, as a function of pH. And over the range of pH 3.6 to 11, there's no change in the kinetic profile, right? There's no discernible lag phase. There's no real change in the nature of this process. However, when we look at the glycosylated molecule at pH 7.4 and above, the kinetic profile is largely similar, right? However, under acidic conditions, similar to water, we see a much slower kinetic profile in these systems. And through mechanisms that we still don't fully understand and are actively investigating, we see that it's not a direct relationship, right? So at pH 3.6, the assembly is actually more rapid than the assembly at pH 5.8, where 5.8 is closer to the pH conditions of the deionized water that we used in the preceding experiments, right? And so some plausible mechanisms that may be at play here would be simply that glycosylation is just changing the solubility of the peptide, right? So the opportunity and availability of the peptide to form prefibrillar aggregates that may nucleate the fibrillization process may be affected by the presence of a single monosaccharide, right? Another logical and possible explanation here. So these peptides are all uncapped at the N terminus. And so the presence of the glycan or the absence of the glycan may be shifting the pKa of the N-terminal amine. It may be changing the charge state of the peptide. In turn, that may change its solubility or its aggregation potential, right? And finally, an area we're particularly interested in is whether or not there are peptide glycan or glycan-glycan interactions that may be at play that are changing the structure of the GQ11 peptide relative to the NQ11 peptide in the unassembled state, and whether those structural perturbations or alterations may be determinants of the rate and pathway of assembly. But nonetheless, to pull this back together in the context of a proteinopathies seminar, is that the takeaway that I want you to leave with from this work is that glycosylation of a non-fibrillizing domain can affect fibril formation, right? And so in my perspective, all glycans should be considered in the context of glycoprotein-based proteinopathies, right? And bringing it back full circle, this is really analogous to that need for glycans in the mucin modeling. Right, we right at the onset and showed really rigorous data from the Twitter sphere demonstrating that the absence of glycans in the AlphaFold modeling completely destroyed the ability to accurately predict mucin structure. And we think that even non-integral, so non-fibrillizing glycosylation sites can have an effect on the formation and progression of proteinopathies. Right. And so to conclude everything that I've presented to you here today, I hope you leave with a sense that glycopeptide fibers can provide the basis for supramolecular biomaterials. I remind those in the group that many of our most abundant natural materials, so things like the plant cell wall and arthropod exoskeletons, are fabricated using carbohydrates, particular polysaccharides, that are involved in assembly processes, and that carbohydrates are the most abundant organic molecules on Earth. Right? And throughout sort of the history of supramolecular biomaterials, molecules like lipids, peptides, and DNA have predominated the workspace, right? And the reason why they have predominated, in my perspective, is that because their behavior is more predictable, they're somewhat simpler, right? And we have some sense of how and why they behave the way that they do. And this is maybe most obviously taken when comparing the rate of growth of the fields like DNA origami to the rate of growth of material systems based on peptides and proteins. More complicated interactions in the world of peptides and proteins has slowed growth in that space. Right? Sugars, despite their natural abundance and their importance in natural biomaterials, have received much less attention. And in part, this is because of the complex, highly challenging synthesis that may come with them, as well as their stickiness and their sort of unpredictable interactions. And so what I want to leave you with is that we feel, right, that sugars deserve their time in the spotlight. That glycosylation is an important part of protein biochemistry, and that there's really ripe opportunity to begin to think about glycosylation in the context of supramolecular biomaterials, particularly with regard to peptides. But as we venture into the space, we can't just consider the function of the glycans. We also must consider the form and the effect that these glycans have on the assembly process and the states that these peptides ultimately adopt. So with that, I'd like to acknowledge the group one more time. Again, this work was led by Dr. Rasuccia and Dr. Zwo, and in large part is now also being carried by Dr. Liu in the lab. Of course, the funding sources who've supported this work all along, and the folks who are here today who took the time out of their day to listen to me talk about this work, and of course the organizers for inviting me to present. So with that, I will gladly take any questions.
Thank you very much, Greg. If you have any questions, please put them in the chat. Oh, I see one. I'll read it to you. How do you think the different structures affects the function of these glycosylated nanofibers and their specificity towards galactans?
Yeah, so that's an interesting question, right? And I think one of the things, so these were the observation of change in structure emerged in the process, right, of sort of developing and building these material systems. And so, one of the things that we would propose is that these aggregation prone or association prone phenomena, right? So where the nanofibers are bundling together is a physical scenario that we would not want in the context of galectin binding, right? And so we would really prefer a scenario in which you have a single nanofiber, right? That's very, very thin and very long. It gives the highest possible density of sugar presentation, while also minimizing sort of non-specific fiber to fiber events. You want to maximize the surface area for protein binding and minimize the fiber-fiber contact surface area. And so I actually have some of the data here from that paper. So you can see, for example, as we change the concentration and the density of the sugar on the fibers, you can either promote or disrupt these alignment events. And so, yeah, I think I think the reason why one of the takeaways is consider both form and function is that we can't ignore the effect that these sugars are having on the structure. It's not as simply as just place a sugar there and it will do what you want. We have to account for the biophysical impact, the structural impact of having those sugars present in the system. So we think that too much sugar is not just bad from that early model from a steric penalty perspective, but also because it may be driving fiber-fiber contact that blocks sites for protein access and protein interaction.
Very good. Thank you, Greg. I'd like to ask a question, if I may take advantage of my position.
Sure.
So I think we can all talk for hours about how carbohydrate modification could affect assembly pathways and peptide structures. I think there's at present too little known. But I want to turn it around. So you mentioned earlier in your talk that carbohydrates are involved in biological recognition, for example, infection, right? So what I'd like to know is how much is known about the structure of carbohydrate assemblies, and how important is the detailed structure of a carbohydrate nanostructure to infection or other biological recognition?
Yeah, so that's a really interesting question, and I would argue that that is a space that is largely in its infancy as well, right? And so one of the challenges there, so most of the oligosaccharides that we work with are really simple structure, and what you will see in naturally occurring oligosaccharide structures are branch points and other bifurcation or trifurcation, right? That will and that each of those arms and that sugar may be terminated in a different way. And so there's a lot of chemical variability in the single glycan molecule level that may have an effect on binding and association, where I think, and that's an area that, necessitates improvements and advancements in chemical synthesis and presentation, but there's emerging knowledge in that space. I think maybe what ties more closely to some of the work here is when we think about how glycans are presented on a folded protein and the accessibility of those glycans and how they can engage with a target. And I would argue that one of the most compelling observations I've seen, at least in that space, is that GP120, which is a coat protein on the HIV virion, right, is a glycosylated molecule, and it has both mannose sugars, but it also has lacnac, so the disaccharide that galectins bind to. And what has been shown is that which galectins can bind to the lacnac on GP120 depend on whether or not GP120 is in its native state or it has been denatured. Right? So the breadth of galectins it can bind to increases when the protein is denatured, but when it's properly folded, there's only a subset of galectins that can bind. So there is on some level, there's a physical encoding by the protein itself that can regulate or dictate which lectin partners can bind, right? And that is it's a really cool problem, right? It's a really ripe space, I think, for investigation. And there's a wide open area in the world of structural biology of understanding, again, how not just the sugar, but the partners around it and the context in which it's presented. How does this dictate binding to a cognate partner? How does it confer specificity or selectivity? And how does that regulate the downstream biological events, right? You would think of, when we think of galectin interactions with the cell surface, there are dozens of glycoproteins on the cell surface that galectins can bind to, right? And so if a galectin is sampling all of those glycoproteins on the cell surface, how does it ever evoke a biological response, right? You think that if it has all of these non-target partners in the background and it's involved in all of these admittedly labile and very weak interactions, how does it ultimately collapse to find a partner that drives the process forward? That sampling time is relatively long and the sampling space is quite large, right? And so there has to be some degree of physical bias, right? That is specifying or selecting for interaction with one partner versus another partner on the cell surface. At least that's a hypothesis that I would put forward. And I think that structural biology is poised to help us answer those questions, but we have to be able to integrate glycans into the models. We have to be able to integrate glycans into the structures, and then we have to be able to look in these very complex environments.
All right. Once again, Greg, you've made us all, our lives much more complicated. So I think we have one more question, a practical question from Carol Hall. Can you attach the sugars on purpose at any point on the peptide that you want?
Yeah, so yes. So depending on how much money you want to spend, we can synthetically couple sugars to any point in the molecule that we would want to couple them. We are working on more sort of cost-effective and federal funding friendly approaches in which we're using naturally occurring enzymes to try to glycosylate tags. So you'd have maybe a 4 or 5 amino acid stretch that would be modifiable as opposed to single amino acid resolution. But yeah, I mean, with the work that we do here, we know where the sugars are located because we're using what end up being quite expensive molecules, starter molecules, in order to synthesize the peptides with the sugars where we want them. So we could change the linker, we could make it longer, we could make it shorter, or we could place it somewhere else in the molecule. That flexibility is at our disposal.
Very nice. Greg, I lied to you. There are more questions. I hope you don't mind.
No, it's fine.
So how do we know, do we know how glycoenvironment differs between cell types?
Yeah, so thanks for another really, I'm just gonna make everyone's life more and more complicated here, right? So, so in terms of particular cell types, there is some understanding and some knowledge of how glycosylation and glycosylation on particular receptors may change with the state of that cell. So for example, there's some knowledge of how, for example, T cells, when they transition from a resting state to an activated state, or when they're biased toward one phenotype or another phenotype, have differences in the glycosylation profile. And that can actually render different degrees of sensitivity to galectin-induced cell death. So for example, regulatory T cells don't usually die in the presence of galectins. In fact, they make quite a number of galectins, whereas activated CD4 or CD8 T cells can die in response to galectin signaling. And that's entirely due to changes in the glycosylation state. One of the things that makes that biology complicated, right, is that glycosylation is a post-translational modification, and it depends on the presence of enzymes there are many, from an engineering perspective, there are a lot of knobs that can be turned there, right? There are the enzymes that put the sugar on, there are the enzymes that take the sugar off, and then there are the enzymes that have to synthesize the precursor molecules for that synthesis to happen. And so if you vary the availability of adders or subtractors, or if you vary the availability of synthesizers, you can drive glycosylation in different directions. But because it's not precisely encoded at the genomic level, it's actually a very difficult state to manipulate reliably experimentally. You can knock out all the glycan, right? Or you could maybe introduce a new glycosylatable site, but to really fine tune that sugar architecture it remains a challenging problem in the context of biochemistry.
Very nice. And I think the last question illustrates how much value you bring to this community. And it is, do your results give rise to any specific mechanism of proteinopathy, specifically amyloid plaque / tau neurofibrillary tangle deposition in neurodegenerative disorders?
Yeah, so I think that so that's an interesting question, and that's what I hope that model systems like ours will start to shed light on. Is, if we this is a question that I think about in some context all the time. I could probably take another hour to talk about what I've been thinking about in this space. But I think the beauty of the synthetic systems that we work with as sort of basic science tools is stripping out the nuance of some of the consensus molecules in proteinopathies, right? So how general is the phenomenon of glycosylating the non-beta sheet domain on, say, amyloid, beta amyloid aggregation or tau aggregation? There's actually some data, a paper a few years ago, I believe it was tau was the molecule that they were working with, and there's an external glycosylation site on tau, and there's some evidence to suggest that glycosylation at that site will perturb the fibrillization process. And so that would dovetail quite nicely into some of what we've seen with our synthetic systems, right? And so, yeah, I think that the direct connection between our synthetic systems and natural systems will really depend on what we can learn from the synthetic systems. And I think it's an opportunity to ask how broad spectrum are these biophysical phenomenon versus how molecule specific are they? And I think in order to know that, again, we have to know more about what is the glycan doing, right? Is it interacting with one or more amino acids within the peptide? So is there an intramolecular interaction that's disrupting fibrillization? Is it an intermolecular interaction? Is it simply a solubility effect? Some of those will translate and some of those will not. And I think that that's why tools like this will ultimately be valuable as a counterpoint to the natural molecule.
Wonderful. Thank you, Greg. So I think we are now done with our questions. And I do want to highlight how much insight we can get on natural protein assembly by looking at engineering systems. And thank you for showing us such wonderful things. Rams?
Thank you very much, Greg. It was a great talk. Thanks, Eitan. It was a great talk as well. And thanks to everybody. Let's open up the session for the entire audience for any questions. I think there were a lot of exciting questions. I got some, but we can keep them for another time. Okay, Rakesh is raising his hand. Let's promote him to the panel. Go ahead, Rakesh. Rakesh? Okay, meanwhile, I can ask a question to Eitan. Could you comment on your sample preparation? You observed dimer, right? So what condition will lead to the dimer formation as opposed to non-dimer formation? Can you comment on that? Do you have any experience on seeing other types of species with your experiments?
So we tried different buffers with different sodium chloride concentration from 300 millimolar all the way to 10 millimolar. And we always see in moles mixed molecular mass at one elution. But when we go to more specific measurements, we are able to see that at lower concentrations, we have intermolecular interactions that could not be explained by higher aggregates at those concentrations. And the masses are never going above the dimer molecular mass. That's what I can say.
Thank you.
So I have another question about the modeling. So we know for Parkinson, BD versus MSA, fibrils are different. Based on your modeling, do you think they divert at the monomer or at other stages of the aggregation process?
So actually, we're checking that these days. And from what I tell you, just observing the smallest oligomer is not enough. I think that what we found specifically on cluster 1 is a compact, stable, abundant dimer among the other possibilities that we cannot account as something that for sure we agree that will drive the known mechanism of aggregation of this protein. And why? Because you expect an exposure of the neck domain and you expect it to gain a beta structure. Well, we got a beta structure, but it's buried. That's one thing. Another thing is that surprisingly, we find an exposed entity beta structure. So this structure might, and this is just a speculation now, might bifurcate between different pathways. One, which is the neck exposed at a later stage, and the other might be off pathway to the known aggregation, but maybe in other aggregation pathways. Taking into account the very long lag phase in kinetics of aggregation, you have multiple possibilities that are futile towards the end goal fibril. And who knows what goes on there? We still don't know.
Thank you.
Yeah, I think it's time to say thanks to everybody and thank you all very much.